Today we'll start with the last part, which is the urinary system. And the urinary system is divided into 4 parts: paired kidneys on each side, ureters from each kidney that join into the storage site of urine, the bladder, and then into the ureter. The urethra, the final part before the excretion of urine at maturation. The major function of the urinary system is the filtration. Therefore, it functions to regulate the acid-base balance and water electrolyte balance, excretion of metabolic waste products, and certain bioactive substances such as many drugs, secretion of renin, which is important to regulate the blood pressure and maintain an optimal glomerular filtration rate, secretion of erythropoietin, which is a growth factor that stimulates the production of erythrocytes in the bone marrow, the formation of the active form of vitamin D, and finally the gluconeogenesis, which is the production of glucose from amino acids. First, starting with the most important part, which is the kidneys. And the kidneys here, which is a bean-shaped structure, range from 12 by 6 centimeters and thickness of about 2.5. It has 2 borders, a concave medial border here and a convex lateral border. The concave medial border here has the hilum, and the hilum is where blood vessels enter. Here, a renal artery entering the kidney and the renal vein exit. It has this dilated part that calls the renal pelvis, comes after the expansion of the ureter here, and then the renal pelvis divide into 2 or 3 structures called the major calyces that form smaller branches called the minor calyces. The whole structures here at the renal hilum is surrounded by adipose tissue. The renal parenchyma here, this is the renal hilum, and this part is called the renal parenchyma, which is divided into 2 parts. The most superficial part here is called the cortex, and a deeper part, and it's called the medulla. The cortex here is darker staining area and contain the site of the functional unit of the kidney, which is the nephron. The medulla here is divided into pyramidal structures or cone-shaped structures called the renal pyramids. The base of each pyramid is at the junction of the cortex and medulla and is called the corticomedullary junction. This is the base of the renal pyramid, and this is the tip. The tip of the renal pyramid here is called the renal papillae, and it delivers the urine filtered into the minor calyces, major calyces, renal pelvis, and then into the ureter. The structure of the renal pyramid with the cortex and the adjacent extension from the cortex called the renal lobe. The cortical extension here between the renal pyramid, these extensions into the medulla is called the renal column. These are the renal column. So renal column, cortex, and the adjacent renal pyramid is called the renal lobe. The whole structure of the kidney is surrounded by a fibrous capsule. The kidney consists from functional units that range from 1 to 4 million, and this number decreases with older individuals, especially in presence of high blood pressure. And those functional units are called the nephrons, divided into 5 parts. This is the nephron part, starting from this part, which is the most important. This section here is representing this part of the kidney, part of the cortex, and adjacent part of renal medulla. Therefore, here, this is the cortex, and this is the corticomedullary junction, and this is the medulla, the renal pyramid. The first part is the renal corpuscle, and this is an initial dilated part to represent a tuft or network of capillaries in the form of loops, and this is very important because this is where the blood filtration occurs. It's always located in the cortex. This is the network of capillaries, and the surrounding capsule together will form the renal corpuscle. So the renal corpuscle consists from the glomerulus. The network of capillaries here is called the glomerulus, surrounded by this capsule, the Bowman capsule, and between the network of capillaries and the capsule is a space where the filtration occurs, the urine, which is the urine, and this filter will pass next into the tubules. The next part after the renal corpuscle here, where the blood filtration occurs, is the proximal convoluted tubule. It is highly tortuous, therefore it is called convoluted, means highly tortuous, and is proximal to the renal corpuscle, therefore it's called proximal convoluted tubule. It is long and also located in the cortex. It has here a short straight part before it enters into the medulla as the lobe of Henle. So the next part, here the lobe of Henle, which is a U-shaped structure, lies within the medulla. The lobe of Henle consists of 2 limbs. Both of them are thin, descending and ascending limb. It continues upward as thick ascending straight tubule that enter the cortex and continue here in proximity to the renal corpuscle and become then convoluted as the distal convoluted tubule. So both the proximal and distal convoluted tubules are located within the cortex, while here the lobe of Henle is located within the medulla. The last part is the starting at the connecting tubules, which are short minor parts that act on linking by linking several tubules. These are short. Here are the connecting tubules. These short tubules will join together and drain into the collecting tubules. The collecting tubules, this one, another one from another site, will join together to form a larger duct called the collecting duct. And then the collecting duct will converge at the tip of the renal pyramid, which is the renal papillae, delivering the urine into the minor calyx here. This is the renal papillae, the tip of the renal pyramid, and this is the minor calyces. Next part of the kidney, the blood circulation, and it is very important because the major function of the kidney is blood filtration. a representation of the blood supply of the kidney, and the renal vasculature here, it is large, closely associated with all elements of the nephron and well organized. Starting from the artery here, the renal artery that enters the kidney here at the medial concave border, that is called the renal hilum. The renal artery divides into two main arteries. Each of them is called the segmental artery. This division occurs in the hilum, and then each of those segmental arteries will divide into further branches called the interlobar arteries. They're called interlobar because they extend between the pyramid here, between the lobes. إن اللوب يتكون من cortex adjoining renal pyramid and the adjacent extension from cortex, which is the renal column, together called the lobe. So this branch that runs between those lobes is called the interlobar arteries. Then this lobar artery will branch here to give an arc-like artery. Therefore, it is called the arcuate artery. And those arcuate artery will give smaller branches called the interlobular arteries, radiating from here. This is the interlobular arteries that radiate from this arcuate artery. And this interlobular arteries is very important because it will form the microvasculature here. This is the microvasculature, the afferent arteriole that will branch into the tuft of capillaries or loop of capillaries that form the renal corpuscle. Magnification of this part here, the nephron part of the cortex again, and the medulla at higher magnification. This is the cortex, this is the medulla, this is the interlobular artery that has branched from this arcuate artery, and this interlobular artery will give branches, which are the efferent arterioles, into the renal corpuscles. The efferent arteriole will enter into the renal corpuscle, divide into or form the network or loop of capillaries, which is the glomerular blood vessels, the capillaries, and then it will exit the renal corpuscle as efferent arteriole. Instead of entering as artery and exiting as vein here in the corpuscle, it enters as arteriole and exit as arteriole. Afferent arteriole entering and efferent arteriole exit. The efferent arteriole that has left the glomerulus here will branch again to form a network of capillaries that is closely related to the proximal and distal convoluted tubules. This branching capillary network is called the peritubular capillaries. Further branches here extend into the medulla, and this is tassel-like bundles of capillaries called the vasa recta, which is closely related with the loop of Henle and also the collecting duct. Those capillaries will deliver the blood into the veins, and the veins here follow the same designation: interlobular veins, arcuate vein, interlobar vein, and then into the renal vein that deliver blood into the inferior vena cava and then into the right side of the heart. The major function of the kidney is filtration, secretion, and absorption. And this is important to control the water electrolyte balance, and that is removed along with the metabolic waste product. This specialized function is carried by epithelial cells lined the nephron within the glomerular cells and the epithelial cells lining the collecting duct system, the proximal, distal tubule, until the last part, the collecting duct. The major function, as said, is the filtration. Also, there is tubular secretion, and secretion means that the substances will move from the lining epithelial cells into the lumen, secreted into the lumen with the filtrate or the urine, while tubular reabsorption means that there are certain substances present in the tubular lumen will be reabsorbed back through also the epithelium, but it will be taken back into the circulation, first through the epithelium, then into the surrounding interstitium, and then into the blood vessels in the interstitium, back into the circulation. So secretion, it will pass into the urine. Reabsorption, it will be taken back into the circulation. The blood filtration is carried by this specialized structure here, the first part of the nephron where the blood filtration occurs, which is the renal corpuscle. The renal corpuscle here begin as loop or tuft of capillaries called the glomerular capillaries, and the glomerular capillaries is contained within a capsule called the Bowman capsule. And the Bowman capsule having two layers or double wall, the parietal and the visceral, and between them is the space in which the filtrate that pass here from the intraluminal space into this urinary space. So the blood will be filtered from the capillaries here into the Bowman space or the urinary space. This special double-walled structure, the capsule, has a parietal layer, which is the outer layer here, aligned by simple squamous epithelium, and a visceral layer or deeper layer here, which is closely enveloping the network of capillaries. These cells that are stellate shaped are the visceral epithelial cells of the Bowman capsule, and they are called podocytes. They are a special type of cells, represent the visceral layer of the glomerular capsule. The blood vessel enter into the renal corpuscle here from afferent arterial branching or loop of capillaries, and then exit through the efferent arterial. And the filtrate from this capillaries pass into this space, the Bowman space or capsular space, and then pass into here into the tubules, starting from the proximal convoluted tubule. The part where the blood vessels enter is called the vascular pole, and the part where here exit into the tubules is called the tubular pole. The parietal epithelium is simple squamous, but as it approaches the proximal convoluted tubule here, it changes into simple cuboidal epithelium. So the proximal convoluted tubule is simple cuboidal epithelium, while the parietal layer of the glomerular capsule is simple squamous epithelium. The important part here of the capsule is the visceral epithelial cells, because they are unusual epithelial cells, has stellate cells called the podocyte, and they are important because they represent the outer layer of the filtration membrane. At higher magnification, you can see the podocyte closely enveloping the capillaries. They are large. This is the nucleus, this is the cytoplasm, and they extend numerous foot processes. These processes that are enveloping the capillaries of the glomerulus, the glomerular tuft. Each podocyte will extend those primary processes, and then each of those processes will extend a smaller processes called the secondary processes are called the pedicels. These small processes called the pedicels, and they cover most of the capillary surface, and below between these pedicels are elongated spaces called the filtration slit. The whole filtration membrane here consists of 3 layers. The innermost layer here is the fenestrated endothelium, the endothelium that line blood vessels. Since the glomerulus consists of network of capillaries, capillaries are blood vessels, so surely the lining is an endothelial cells. The characteristic of endothelial cells here and the filtration membrane that it has fenestration, these holes or openings. These openings help the filtrate to pass through the endothelial cells. And the middle layer between the endothelial cells and the overlying podocyte is a thick glomerular basement membrane, and it is the most important part of the filtration membrane. It's formed by fusion of the lamina of endothelial cells and podocytes. And finally is the outer layer of the filtration membrane, which is the visceral layer of the glomerular capsule that is called the podocytes. This is an electron microscope image of the glomerular filtration barrier, and we can see this is the high magnification, this is low magnification, and this is high magnification. This section shows this is the lumen of the capillary. These are blood cells within the lumen of the blood vessel, the capillary. The lining of the capillary here are endothelial cells. This thick gray line is the glomerular basement membrane, and this is the outer cells. These large cells are the podocyte. This is the nucleus of the podocyte, and this is the cytoplasm of the podocytes. These minor structures are the pedicels, the secondary small processes of the podocytes. At higher magnification, this section is magnified here to see the 3 layers of the glomerular filtration barrier, the endothelial cells. This is an endothelial cell. This is the capillary lumen, and this is the lining of the capillaries. This space between the adjacent endothelial cells is the pores or the fenestrations that is present between the endothelial cells, and it helps the filtrate to pass through the barrier. Next here is the glomerular filtration membrane, formed by fusion of the basal lamina from endothelial cells and the podocyte, and these are the podocytes. This is the pedicels of the podocyte, and the spaces here between the pedicels. نرجع للصورة بالدياجرام أوضح. هذا هو slit diaphragm. This is the secondary processes. This is the primary processes, and spaces between the secondary processes is the filtration slit or slit diaphragm. Slit diaphragm here is a zipper-like structure formed by tight junction. This is the slit diaphragm. The tight junctions formed by nephrin and other proteins, glycoproteins, projecting from the cell membrane on each side here of the filtration slit. The filtration again occurs through the fenestration or the holes of the endothelium. And this is important because it blocks all the blood and platelets and large cells in the blood. The glomerular basement membrane, which is the most important part of the filtration barrier because it separates the blood and the capsular space, and it holds the most organic anions and the large proteins, preventing them from passing into the urine. And lastly, the filtration slit, which is formed by parts formed by the pedicels or slit diaphragm, This also restricts small proteins and organic anions. Normally, about 20% of the plasma volume will enter the glomerulus is filtered into the capsular space. The initial filter rate has a chemical composition that is quite similar to the plasma, but the difference that it has a very low protein level because most of the plasma proteins is prevented from being filtered. This is another picture of the glomerular filtration barrier. This is the large blood cells, leukocytes, platelets, red blood cells. They are all too large to pass through this filtration barrier. In addition to the proteins of large molecular weight, they cannot pass through the filter, while smaller proteins and organic molecules, glucose, amino acids, all pass first through the fenestration here of the endothelium, then through the glomerular basement membrane, which block the larger proteins, and finally the slit diaphragm that lies between the pedicels, the secondary photoprocesses of the visceral glomerular epithelial cells, the podocytes. And also this is important to control the passage of small proteins. So the large proteins are mainly prevented here at the glomerular basement membrane. The last part of the renal corpuscle here is called the mesengium. And the mesengium is a supportive structure, consists of cells, mesengial cells, and matrix called the mesengial matrix. The major function of this part is supportive, and it fills the gap where the podocytes are not covering the capillaries. In addition to its main function, the physical support, it has some contractile ability to control the blood flow in responses to blood pressure changes. Also, it has a phagocytic function for certain protein molecules that adhere to the glomerular filter, and secretion of several cytokines and prostaglandins. The next part of the urinary system are the tubules and the ducts. Starting from the proximal convoluted tubule, and as I said, it's called convoluted because of highly tortuous structure, and proximal because its proximity to the renal corpuscle here. The first part after the renal corpuscle is the proximal convoluted tubule. The major function of this part is reabsorption and secretion. Reabsorption is very important because it's most of the reabsorption, about 50% of the tubular reabsorption is carried by the cells of the proximal convoluted tubules. To include water, electrolytes, organic nutrients such as amino acids, glucose, all of these are reabsorbed in the proximal convoluted tubule. The other function is secretion, and the secretion means its passage through the epithelial cells of the proximal convoluted tubule and pass into the filtrate, into the urine. The other function is the production of the active form of vitamin D. This function is carried in the proximal convoluted tubular epithelial cells. Histologically, the cells lining the proximal convoluted tubule is simple cuboidal epithelium. It has a central nucleus here, and the characteristics of proximal convoluted tubule that has a reflection on its functional influence is the presence of abundant mitochondria, and abundant mitochondria will give this bright eosinophilic or acidophilic cytoplasm, and it means also there is an abundant production of ATP energy that is required for its major function, which is reabsorption, the active transport. Also, it has a tall, prominent brush border, which is important to facilitate reabsorption. And this section shows the renal corpuscle, this network of capillaries, capillary tuft. The surrounding capsule, this is the parietal epithelial cells, and this is the capillaries, and the space between them, between the and the capillaries here is the urinary space. So this is the structure of renal corpuscle. The adjacent structures here, all of them are tubules. Those ones who are bright eosinophilic cells here, this eosinophilia is imparted by mitochondria, is the proximal convoluted tubule. All these are proximal convoluted tubules. Those cells, the smaller ones, much less acidophilic, are the distal convoluted tubule. Next part of the tubules are the loop of Henle. This is the loop of Henle, and it begins after proximal convoluted tubule. It has a thick straight part, runs here and then enter the medulla, continue as the loop of Henle, which is U-shaped, containing a thin descending and a thin ascending limb. Both of them is lined by simple squamous epithelium, unlike the proximal and distal convoluted tubules, which are lined by simple cuboidal epithelium. Therefore, the major function here by the loop of Henle is the passive transport and not an active transport. After the thin ascending limb of the loop of Henle, here it becomes a thick ascending limb, which is again lined by simple cuboidal epithelium with many mitochondria and an active role in the transport. It will extend upward, entering into the cortex, up to this part of the distal convoluted tubule, just next to the glomerulus, just next to the afferent and efferent arterioles. This special part of the distal convoluted tubule is called the macula densa, together with the cells of the afferent arteriole, forming the juxtaglomerular apparatus, which is important in controlling the blood pressure and the glomerular filtration. The ascending limb here of the nephron entering the cortex after the formation of the macula densa here to become tortuous and continue as distal convoluted tubule. The major function of the distal convoluted tubule is also tubular reabsorption, but compared with the proximal convoluted tubule, the reabsorption here is much less. The lining of the distal convoluted tubule and it's involved in the tubular reabsorption is simple cuboidal epithelial cells, and as mentioned, it's different from the proximal convoluted tubule. نرجع للصورة اللي قبلها هذه. Here, the cells are smaller, less acidophilic because it contains less mitochondria and less or sparse short microvilli, and this means it's involved in less tubular reabsorption. The lumen here is also appears less empty, while here more secretions or more content in the lumen. The whole function here of the reabsorption or absorption of the sodium in the distal convoluted tubule is controlled by aldosterone, which is secreted from the adrenal gland. The next part of the distal convoluted tubule here is called the juxtaglomerular apparatus. The distal convoluted tubule, as it enters the cortex and approaches the renal corpuscle, it it comes into contact with the afferent arteriole. The site at this contact here will form what is called the macula densa. The cells of the macula densa will change morphologically into taller columnar epithelial cells, and they are more closely packed. Here, the cells are located into the distal tubule and comes into the contact of the cells of the afferent arteriole. The cells of the contact with the macula densa cells in the afferent arteriole is called the juxtaglomerular cells. The cells of the macula densa and juxtaglomerular cells are called the juxtaglomerular apparatus. And this apparatus functions by the autoregulation of the glomerular filtration rate through controlling of blood pressure. So, if there is an elevated blood pressure, this means increase in the capillary blood flow or blood pressure, and then again, this will increase the glomerular filtration rate. Higher glomerular filtration rate means there is higher concentration of luminal sodium and chloride. This is monitored through receptors in the distal convoluted tubules called osmoreceptors here. Sense any change in osmolarity, especially the sodium, and if these receptors are stimulated, it will increase levels of releasing certain substances such as ATP, vasoactive substances. These substances released from the macula densa will trigger contraction in the afferent arteriole. This contraction will lower the glomerular pressure here and decrease the glomerular filtration rate. The opposite happens when there is a drop in the blood pressure. The juxtaglomerular cells here will sense also changes in the blood vessel in a similar way, and in response to changes in the blood vessel, the juxtaglomerular cells will release the renin. And renin is an protease enzyme, cleave angiotensinogen into angiotensin 1, and then angiotensin 2, which is the potent active part that function to vasoconstriction of the blood vessels, and by vasoconstriction blood vessels, it will change the blood pressure and the flow rate into the glomerular capillaries. These changes are sensed by special receptors called the baroreceptors. Next part is the collecting duct, and it is the last part of the nephron. Again, starting with the short, straight connecting tubules that are collecting the urine into the collecting tubules, and the collecting tubules will from several nephrons join together into forming a large duct called the collecting duct. The lining here is simple cuboidal epithelium, but in the largest part of the collecting duct, as it extends or progresses deeply into the medulla, it will change into more columnar epithelial lining. So it begins as connecting duct, connecting tubule, and then collecting duct. This large duct, the collecting duct, will merge and become more straighter and larger, more epithelial, more columnar lining, until it reaches the tip of renal pyramid, forming the papillary duct. At the tip of the renal pyramid, which is called the renal papillae, therefore the duct at this side is called the papillary duct, because it's site at the tip of the renal medulla that is called the papillary duct. And this function to deliver the urine into the minor calyx. The major function of the collecting ducts and tubules is the final concentration of urine before its passage into the calyces, pelvis, and then into the urethra. This section shows a deep part of the tubular and duct system. So this part here shows the loop of Henle and the collecting ducts. The section here is from the medulla. This part is the thick ascending duct, which forms the initial part of the distal convoluted tubule here. Thick ascending part. And you can see here the line is simple cuboidal epithelium, bright acidophilia, because its function in the active transport. And these thin structures lined by simple squamous epithelium are the loop of Henle. The thick ascending, the thin ascending, and the thin descending limbs of the loop of Henle. These large structures here are the collecting ducts. Here it appears to be more columnar and have unusually distinct membrane compared to the ascending ducts. The lining epithelial cells of the ducts and tubules consist of large pale cuboidal cells called the principal cells that will change into more columnar morphology as it goes distally. The major function is the regulation of water and electrolyte reabsorption and secretion of potassium. This is carried under influence of antidiuretic hormone from the posterior pituitary gland. In addition to principal cells, it has a fewer population of darker staining cells called the intercalated cells, and they has two major functions: maintaining acid-base balance through controlling hydrogen ion and bicarbonate, and reabsorption of potassium. This is and, not by, and reabsorption of potassium. The other parts of the urinary system other than the kidney, and the kidney is the most important, have the major part in the urinary system because it carries the function or the aim of the urinary system, which is blood filtration. Next is basically a transport system: the ureter, bladder, and urethra. Ureters carry the urine into the bladder, the temporary storage site for urine, and then its final passage way through the urethra. The urine passes from the renal pelvis into the ureter, then into the bladder and the urethra. Starting from the first part, which is the ureter. Here are the ureters. This is kidneys, and a ureter from each kidney will pass down and drain into the urinary bladder, and then from the urinary bladder here is the urethra. First, the ureter here, you can see the wall of the ureter, which is similar to the calyces, renal pelvis, and has mucosa that consists of transitional epithelium or called urothelium and underlying lamina propria, and a muscular wall. And then the outer is the adventitia. There is no submucosa in the ureter. The lining muscular wall becomes thicker here near the urinary bladder, and the thickness here is important to contraction and help to carry down the urine into the urinary bladder. The mucosa of all these organs, the urethra, bladder, and ureters, is lined by a similar type of epithelium, which is a unique stratified epithelium called the urothelium. This is the urothelium, or called the transitional epithelium. And all these organs, the urothelium consists of three layers. The basal layer, which is small basal cells resting on a very thin basement membrane here. This line are the small basal cells. And then an intermediate layer, which are cuboidal to low columnar cells, and the most superficial outer layer, which are large bulbous cells called the umbrella cells, and they are highly differentiated and they function to protect the epithelial cells or the urothelial cells from the cytotoxic effects exerted by the urine contained within the lumen of those organs. Also, this is a section of the ureter. Here we see the outer layer adventitia here, the thick muscularis of the ureter that help to move the urine toward the bladder and produce prominent mucosal folds here when the lumen is empty. So this is an empty ureter. These folds will become less when the ureter is filled. On the bladder, the lining urothelial cells become more specialized. This is the bladder lining, and this is the lining urothelium or transitional epithelium underlying lamina propria, the submucosa, the muscular wall, the inner, middle, and outer muscularis propria, and the outer adventitia. The three muscular layers here are poorly delineated, but they form a thick muscular wall for the urinary bladder. The specialized feature of the urinary bladder epithelium that the lining umbrella cells, the most superficial layer of the urothelium, has an extensive intercellular complexes. Between these cells there are complexes, and they are formed by hinged areas. These hinged areas formed by proteins called uroplakin. And these hinged areas help to change in the morphology of the lining urothelium to highly folded when empty and become more extended here, and change movement in the hinged areas will change its morphology into flattened appearance when the bladder is filled. The folds here increase the surface area by folding the membrane at the hinged area and internalizing the plaques. The urethra, which is the last part of the urinary system, is a tube that functions mainly to carry the urine to the exterior, and also as other part it has longitudinal folds. The urethra is different in men and women, and one of the differences that the ejaculatory duct will join the urethra at its initial part in the prostate gland. This is the male urethra, which is longer and divided into three different parts, starting from the prostatic urethra here. This is the seminal vesicles, the duct from the vas deferens, and the seminal vesicles from the ejaculatory duct, and the ejaculatory duct joins the prostatic urethra here in the prostatic gland. The first part of the male urethra runs through this gland, the prostatic gland. Therefore, it is called prostatic urethra. It's about 3 to 4 centimeters, and the lining here is similar to the bladder and the ureter, and therefore it is a urothelium. The next short part runs between the first and second part, and it's called the membranous urethra. It passes through the external sphincter of striated muscles. The lining here is different. It's not urothelium as in the prostatic urethra, urinary bladder, and the ureter. The lining here is columnar, stratified and pseudostratified columnar epithelium. The last part here is called the spongy or penile urethra. As its name implies, penile means it runs through the center of the penis. We have seen this structure with the male genital system, and it's the longest. That's about 15 centimeters, lies within the corpora spongiosum of the part of the erectile tissue, and the lining is also columnar epithelium like the membranous urethra, pseudostratified or stratified columnar epithelium, but here near the end it changes into stratified squamous epithelium. In women, the urethra is mainly a urinary organ, while in the man it's also an ejaculatory organ. The length is small, 3 to 5 centimeters only, therefore the incidence of urinary tract infection is higher in the woman because of the short tract, easier access into the urinary tract system by bacteria. Also, the anatomical position of the female urethra is close to the anus, and this helps the bacteria to gain easier access into the urinary tract. The lining is also transitional epithelium or urothelium, but as it goes further, it changes into keratinized stratified squamous epithelium similar to the skin of the labia minora. And in both parts, the urethra is surrounded by a striated muscle sphincter, external sphincter. This contraction from this sphincter controls the excretion of urine through the urethra. This is a section of the urethra. Here you can see the mucosa is also folded, lining urothelium. بما أن lining here urothelium or transitional epithelium, for this is either a proximal part of female urethra or the prostatic urethra. At higher magnification here, you can see the thick epithelial lining of the urethra. واضح؟ طلاب، عندكم سؤال على urinary system؟ كل شي ما فاهمينه أعيد لكم إياه؟ أوكي.
واضح دكتور.
تمام، شكراً.